Plants surround almost every moment of daily life. They line streets, fill rooms, shape landscapes, and quietly regulate the air moving in and out of your lungs. They are so constant that they fade into the background, treated as scenery rather than participants. And yet plants are never passive. Every leaf, every root tip, every growing stem is continuously responding to the world around it. Light shifts, temperature changes, nearby organisms move closer or farther away. Water becomes scarce or abundant. The chemistry of the air subtly changes. None of this goes unnoticed. Plants do not see, hear, or think in any familiar sense. They have no brains, no nerves, no conscious awareness. But they sense. They integrate information. They adjust their behavior moment by moment, often more precisely than animals that appear far more active. To understand plants properly, it helps to let go of human intuition. Experience does not have to feel like anything. It does not need awareness, emotion, or intention. In biology, experience can simply mean being shaped, informed, and guided by the world through continuous interaction. From this perspective, plants are immersed in a rich sensory environment, a world defined not by sights and sounds, but by gradients of light, pressure, moisture, chemistry, and time. A world that moves slowly but never stops changing. Tonight's journey is an attempt to step into that unfamiliar perspective, to explore what the world looks like when it is experienced without eyes, without thoughts, and without urgency. Not to romanticize plants or turn them into something they are not, but to understand them more accurately and perhaps more respectfully. Because once you begin to see how plants experience their surroundings, it becomes harder to see the living world as silent or inert ever again. Hello, and welcome to Quiet Science. If you enjoy calm, careful explorations of how reality actually works, consider liking the video and subscribing to the channel. And let me know in the comments where you're watching or listening from tonight, whether that's surrounded by plants or far away from them, in a place where their quiet work is still shaping the world around you. Plants are often treated as scenery, green, patient, decorative, something that sits politely in the corner of the world while everything else gets on with the real drama of life. But a plant is not a painted backdrop. A plant is a living system that spends every moment receiving information, comparing it against internal conditions, and adjusting what it does next. It cannot run, it cannot shout, and it cannot blink in surprise, yet it is continuously engaged with the outside world. Light changes, humidity shifts, soil tightens, a leaf is bruised, a neighbour grows taller, a fungus brushes past a root tip, carbon dioxide rises and falls with the passing day. None of this is irrelevant to a plant. None of it is ignored. The question is not whether plants interact with their environment. That part is easy. The question is what it means in biology to say that a living thing experiences the world. In everyday language, experience sounds like something felt. It sounds like an inner movie, a private point of view, the warmth of sunlight on skin, the sting of cold air, the sharp edge of pain, the comfort of a familiar room. Humans are surrounded by experience in that sense, and it is natural to assume that experience always comes with awareness. Biology is more careful. In biology, experience can be framed in a way that does not require consciousness, emotion, intention, or a mind. A working definition can be simpler and still powerful. A living system experiences its environment when it can sense conditions, integrate information, and respond in a way that changes its future state. Sensing means detecting a signal. That signal might be light, a chemical gradient, pressure, temperature, water availability, or damage to tissue. Integration means combining signals so that a response is not a single reflex, but an outcome shaped by context. A plant does not simply open its stomata because light is present. It considers water status, carbon dioxide levels, humidity, temperature, time of day, and internal hormonal cues. Integration is where raw sensation becomes a meaningful pattern. Response means acting on that integrated information. In plants, action often looks like growth direction, changes in chemistry, shifts in metabolism, reinforcement of tissues, or the activation of defense pathways. It is not dramatic in the way an animal response can be. It can be slow, subtle, and extraordinarily effective. This is a kind of experience that does not have to feel like anything. It is not a claim about inner life. It is a claim about structured interaction with the world. A useful minimal framework here is stimulus and response. Something happens outside the organism detects it, and something changes inside the organism that affects what it does next. This framework is deliberately modest. It does not declare that a plant suffers or hopes or fears. It simply acknowledges that the world presses in on living tissue, and living tissue pushes back in organized ways. Even this minimal framing can be surprising because it forces a quiet shift in perspective. Instead of asking whether a plant is aware, it becomes possible to ask what signals it can detect, how those signals are processed, and how behavior emerges from the system as a whole. That shift matters because human intuition is not built for plants. Human senses are fast. Human perception is tuned to motion, edges, threats, faces, and voices. Human time is measured in breaths and heartbeats. A plant lives on a different tempo, and it uses different channels of information. The world that matters most to a plant is not the world of sound and sight. It is a world of chemistry, moisture, pressure, light quality, and slow repeating cycles. Plants are ideal for testing human assumptions about experience because they remove the usual shortcuts. There is no face to read, no eyes to meet, no obvious moment of choice. Yet the evidence of sensing and responding is everywhere if the right questions are asked. A root tip grows toward water. A seedling bends toward a window. A vine finds support and tightens around it. A leaf changes its chemistry after an insect bites. A tree adjusts the timing of bud burst based on photoperiod and temperature history. Each of these is an interaction with the world that is shaped by information. This is where caution becomes important, because the topic attracts strong opinions. Some people insist that plants must have consciousness, because they behave in complex ways. Others insist that plants are basically machines, because they do not have neurons or brains. Both positions often come from the same habit, turning complexity into a verdict about inner life. The calmer approach is to stay with what can be measured. Plants detect signals, plants integrate signals, plants respond. These are the claims that can be explored without drifting into fantasy, and without stripping plants down into something lifeless and inert. The aim is not to smuggle human feelings into a leaf, and it is not to reduce a living organism into a silent object. The aim is to understand what plants actually do, and what the world looks like when it is filtered through plant biology. So the ground rules are simple. The focus stays on mechanisms and evidence. When uncertainty exists, it is acknowledged plainly. When consensus exists, it is stated clearly. And when words like experience or communication are used, they are used carefully in the biological sense. Information, integration, response. With that in place, the next step is to confront the most obvious objection. If plants experience the world in any meaningful sense, how can they do it without neurons? Neurons are the celebrity cells of the animal kingdom. They are fast, excitable, and dramatic. They generate electrical impulses, they send signals across synapses, and they build networks that support perception, memory, and movement. In many animals, neurons are concentrated into a brain, which allows rapid integration of sensory information and coordinated responses. A typical neuron receives input through branching dendrites, integrates that input in its cell body, and sends an output signal along a long axon. At the end of that axon, neurotransmitters cross a synapse to influence the next cell. This structure makes speed possible. it also makes modular processing possible, where different neural circuits handle different tasks. Plants do not have neurons, they do not have synapses, they do not have a brain, and yet plants still coordinate across long distances. They still integrate multiple signals. They still respond in ways that look purposeful, even when the word purpose is used cautiously. The reason is that neurons are not the only way to build an information system. Plants solve the same fundamental problem in a different style. Instead of relying on a central command center, they distribute sensing and response throughout their tissues. Instead of acting quickly, they often act continuously, adjusting in small increments that add up to major changes over hours, days, or seasons. This distributed approach begins at the cellular level. Every plant cell is enclosed by a membrane that controls what enters and leaves. Embedded in that membrane are proteins that act as receptors and channels. They detect chemicals, shifts in ion concentrations, changes in pressure, and damage signals. When these receptors are triggered, they start cascades of internal changes. Calcium levels can rise inside the cell, gene expression can shift, hormones can be synthesized, metabolism can be redirected. That is sensing and response, right there, without anything resembling a neuron. Integration happens when signals spread between cells and tissues, and when different signals converge on shared pathways. A plant does not need a brain to integrate information if integration is happening across a network of living cells that constantly exchange chemical and electrical messages. One of the foundational figures in understanding plants as active physiological systems was Julius von Sachs. In the 19th century, Julius von Sachs helped establish experimental plant physiology as a rigorous discipline, focusing on how plants grow, how they transport water, how they respond to light, and how their internal processes can be studied with careful measurement rather than speculation. That historical shift matters. It moved plants from being treated as botanical curiosities to being treated as organisms with measurable functions. It also laid groundwork for the modern view that plant behavior is not mysterious in the supernatural sense, but it is often surprising in the biological sense. Plant coordination works through tissues rather than nerves, and through signals that travel in multiple ways. Two main internal languages appear again and again in plant biology: chemical signaling and electrical signaling. Chemical signaling is the more familiar one. Plants produce hormones, small molecules that influence growth, development, and stress responses. These hormones can be transported through the plant's vascular system, move cell to cell, or act locally. Plant hormones are not emotions in liquid form. They are coordination chemicals. They help one part of the plant inform another part of the plant about what is happening and what priorities should shift. A change in water availability in the soil can lead to chemical signals that alter stomatal opening in leaves. An insect bite can trigger chemical cascades that increase defensive compounds, not only at the bite site but across distant tissues. A shaded leaf can influence growth patterns in other regions by altering hormone distribution. Chemical signals are slower than neural impulses, but plants do not need to win races. They need to survive seasons. Electrical signaling is less widely appreciated, but it is real and measurable. Plant cells maintain electrical gradients across their membranes, just like animal cells. When certain channels open or close, the electrical state of the cell changes. Under some conditions, changes in electrical state can propagate along tissues, allowing information to travel faster than simple diffusion. These electrical signals can coordinate responses across the plant, especially during rapid events like wounding. Electrical signaling in plants does not create a nervous system. It does not imply a brain. It is better understood as a network property of excitable living tissue, where electrical changes can act as signals alongside chemical ones. And then there is the plant body plan itself, which shapes experience in a way animals do not always share. A plant is modular. It grows by adding repeated units: leaves, stems, roots, branches. It can lose parts and continue. It can redirect growth. It can rebuild. In many cases, It does not have a single vital organ, whose loss means instant death. This modular structure supports distributed sensing and distributed response. It also means that a plant's self is not concentrated in one location. There is no single seat of control. The plant is, in a sense, an organized negotiation among its parts. Roots report on water and nutrients. Leaves report on light and carbon dioxide. Stems and vascular tissues serve as transport and communication routes. Reproductive structures respond to season and internal energy status. The outcome is a whole organism adjusting itself continuously without needing a command center. This is where the idea of tissue-level coordination becomes important. In animals, coordination is often imagined as a brain sending orders. In plants, coordination is often closer to a network finding balance. Not a conscious balance, but a physiological one. Growth, defense, repair, and reproduction compete for resources. When conditions change, the balance shifts. A plant in full sun with abundant water can invest in growth. A plant under drought stress might close stomata, slow growth, reinforce tissues, and shift metabolism. A plant facing herbivory might divert energy into defense chemicals and wound repair. These shifts are not moral choices. They are structured outcomes of signals interacting within the plant's internal networks. Even at this early stage, one quiet conclusion begins to form. If experience is defined as sensing, integrating, and responding, then plants experience the world constantly, not as conscious witnesses, not as thinkers, but as living systems tuned to their surroundings. And once that idea is accepted, another becomes easier to see. A plant does not need eyes to take light seriously. It does not need ears to detect vibration or touch. It does not need a brain to register danger. It needs reliable signals, and it needs ways to translate those signals into changes in physiology and growth. Plants have exactly that. The world, to a plant, is not a theatre of sights and sounds. It is a landscape of gradients and cues. It is a slow weather of chemistry and pressure. It is time, written into light cycles and temperature histories. It is damage, translated into hormones and electrical pulses. It is opportunity, expressed as water, minerals and open space. And in that world, the plant does what it has always done. It pays attention in the only way it can, not with thoughts but with tissue, not with intention but with integration, not with a single decisive moment, but with an unbroken chain of small adjustments that over time can reshape an entire organism. A human can walk into a room and immediately feel the difference between warmth and cold. A plant cannot do that in a single instant, but given enough time, it will record the difference in growth patterns, water use, gene expression, chemistry, and structure. It is a different kind of awareness, a biological awareness, a measurable one, and it sets the stage for the next step, where the plant's senses begin to come into focus, one channel at a time, revealing how much information is flowing through a world that looks, to human eyes, perfectly still. Light is the fastest information on Earth. Before a root ever touches water, before a leaf ever feels the drag of wind, before a stem ever stiffens in response to weight, light has already arrived. It has already changed. It has already hinted at what kind of day this will be, what kind of season is approaching, and whether nearby plants are about to become competitors. To human senses, light is mostly a way to see. To a plant, light is a message. Many messages layered on top of each other, arriving continuously from every direction. Plants do not need eyes to make sense of light, because they do not interpret light as an image. They interpret it as data, direction, intensity, color balance, duration, rhythm, the difference between open sky and shade, the presence of neighbors, the approach of winter, the beginning of spring. The first key idea is that plants have photoreceptors, molecules that change their behavior when they absorb light. These photoreceptors are not general purpose. They are tuned to different parts of the light spectrum and different kinds of information. One major group is phytochromes. Phytochromes are especially sensitive to red light and far red light. That distinction is not a trivial detail. Sunlight contains both, but when light passes through leaves, the balance shifts. Leaves absorb more red light for photosynthesis and allow more far red light to pass through or reflect. That means the ratio of red to far red light becomes a kind of neighborhood signal. A plant can detect whether it is surrounded by other leaves, even before it is fully shaded. Another group is cryptochromes. Cryptochromes respond strongly to blue light, and they are deeply involved in growth regulation and timekeeping. They help plants track the quality of daylight and adjust development accordingly. Then there are phototropins, also tuned to blue light, but especially important for the sense of direction. Phototropins help a plant detect where the light is coming from, so it can lean, adjust, and grow toward it. Phototropins are central players in phototropism, the slow bending of stems and shoots toward a light source. Phototropism looks almost like curiosity when seen in time-lapse. A seedling on a windowsill bends toward the brightest patch of sky as if it has decided to go looking for it. The underlying mechanism is more elegant than any metaphor. Directional light triggers unequal distribution of a hormone called auxin across the growing tissue. Auxin influences cell elongation. When auxin becomes more concentrated on one side of a stem, cells on that side elongate more. The stem curves, the plant changes its angle. Over time, growth becomes a steering wheel. This is not a single action, it is a continuous correction. A plant does not point once and stop. Light changes as clouds pass. A room brightens in the morning and darkens in the evening. A neighbour's leaves shift in the wind. The plant responds not with a sudden turn, but with gradual rebalancing, adjusting growth rates on different sides so the body slowly reorients. Long before modern molecular biology, careful observation made it clear that something like light perception was happening inside plants. Charles Darwin and Francis Darwin studied plant movements in the late 19th century and explored how seedlings respond to light. One famous result was that the tip of a seedling seemed essential for sensing light direction, while the bending occurred lower down. The tip detected the cue, and the rest of the plant carried out the movement. That is an early glimpse of distributed sensing and signal transmission, where information is detected in one region and used elsewhere. Light direction is only one part of the story. Intensity matters too, not just as energy for photosynthesis, but as a signal of exposure, stress, and opportunity. High light can mean abundant energy, but it can also mean risk. Too much light can damage the photosynthetic machinery. Plants respond by adjusting pigments, changing leaf orientation, opening or closing stomata to balance carbon dioxide intake and water loss, and activating protective pathways that dissipate excess energy safely. Low light can mean shade, and shade is not merely dim. Shade has a specific signature. Under a canopy, red light drops more than far red light. A plant using phytochromes can detect this shift and interpret it as competition. This triggers what is often called the shade avoidance response. Shade avoidance is a suite of growth changes. Stems elongate, leaves may become positioned differently. The plant invests in reaching upward, trying to escape the shadow. It can become taller and thinner, chasing light as if light were a rumour spreading above the canopy. This strategy is not free. A taller, thinner plant can be less stable, more vulnerable to wind, and more likely to suffer water stress. Shade avoidance is a trade-off, a calculated risk expressed through chemistry and growth patterns rather than deliberation. In dense vegetation, this becomes a quiet arms race. Each plant is reading the same signals. Each plant is responding. The canopy becomes a dynamic battlefield of information, where the colour balance of light is the early warning system. Day length adds another layer. Plants can detect the duration of light and darkness across a 24-hour cycle and across the year. This is not simply noticing that it is bright or dark. It is measuring how long those states last. A few minutes of difference in day length can matter, because for many plants it is the most reliable seasonal cue. Temperature can jump unpredictably, rain can come and go. But photoperiod, the length of day, changes with the calendar in a way that cannot be fooled by a warm afternoon in late winter. To treat day length as information, a plant needs an internal clock. It needs a way to compare the external world to internal time, so that light arriving at a particular internal phase means something specific. This is where light sensing begins to merge into timekeeping. Light, in other words, is not just a signal. It is also the standard by which plants build their sense of time. A plant living outdoors experiences light as a repeating pulse. Dawn, midday, dusk, night, dawn again. Even in a stable environment, these cycles are relentless. They shape metabolism, growth, repair, and defense. They also shape anticipation. Because one of the strangest and most revealing facts about plant life is this: plants do not only react to the world as it is, they also prepare for the world as it is likely to be. That preparation is guided by circadian rhythms. Circadian rhythms are internal biological clocks that run with a roughly day-long period. In plants, these rhythms influence gene expression, hormone levels, photosynthetic efficiency, stomatal opening, growth timing, and countless other processes. A plant's body is not doing the same thing at every hour. It is changing state across the day, even if the environment is held constant. The existence of plant circadian rhythms becomes obvious in simple experiments. If a plant is placed under constant light, many of its behaviors still show daily oscillations for a time. Leaf positions may rise and fall. Gene expression patterns can continue to cycle. The plant carries an internal schedule. An early figure associated with the study of biological clocks in plants was Erwin Bunning. In the 20th century, Erwin Bunning investigated plant rhythms and helped develop the idea that internal clocks are central to photoperiodism, the ability of organisms to respond to the length of day and night. This work helped clarify that plants do not simply measure light duration as a passive stopwatch. They interpret light duration through the lens of an internal rhythm. That internal rhythm needs calibration because an internal clock that drifts away from the real day-night cycle becomes useless. Calibration happens through entrainment. Entrainment is the process by which external cues set and reset the internal clock. Light is one of the most powerful entraining signals. Temperature is another. When dawn arrives, photoreceptors respond. When temperatures rise and fall, metabolic rates shift. These signals nudge the clock back into alignment, so the plant's internal time stays connected to the outside world. This calibration is not a one-time event. It is constant maintenance. A plant is always balancing its own internal rhythms against changing conditions. With entrainment, circadian rhythms become not just internal oscillations, but a way of experiencing time. Time is turned into chemistry, and chemistry is turned into behavior. A simple example is stomatal timing. Many plants open stomata during the day to take in carbon dioxide for photosynthesis, and close them at night to reduce water loss when carbon dioxide uptake is not useful. That seems like a direct reaction to light, but the timing can be more subtle. In some cases, stomata begin to anticipate dawn, shifting behavior before the light actually changes. That anticipation is the clock speaking. Growth timing can show similar patterns. Some plants grow more at certain times of day or night, coordinating cell expansion with water availability, temperature, and internal metabolism. Repair and defense pathways can cycle too, because threats are not evenly distributed across time. Herbivores may be more active at certain hours. Temperature stress may peak during midday. A plant that aligns its physiology with predictable timing gains an advantage. Timekeeping becomes even more consequential when it expands beyond daily rhythms into seasonal timing. Photoperiodism is the ability of plants to respond to day length in ways that influence flowering, dormancy, bud formation, and seasonal transitions. The key point is, that plants often use night length as the decisive cue. For many species, the length of uninterrupted darkness can be more informative than the length of light. This is why a brief flash of light during the night can disrupt flowering in certain plants. It breaks the night into segments. The plant's internal system interprets this as a different season. What looks to human eyes like a harmless light flicker can become a strong biological message. Seasonal timing is in a sense long-form experience. It is the plant's record of changing conditions, encoded in gene regulation, hormone balance, and developmental decisions. Gene regulation might sound abstract, but it is one of the most practical tools in biology. When a plant shifts seasons, it is not merely behaving differently, it is changing which proteins are produced, which enzymes are active, and which developmental pathways are open or closed. These changes alter the plant's structure and priorities. A plant approaching winter may initiate dormancy. Dormancy is not simply sleep. It is a physiological state with altered metabolism, protective chemistry, and reduced vulnerability to freezing. It may involve forming buds with protective scales, storing resources, and preparing tissues to handle cold stress. A plant approaching spring may begin to break dormancy. That can involve complex decision-making based on accumulated cold exposure, rising temperatures, and day-length cues. Many temperate plants require a period of cold, a process often called vernalization, before they will flower. The cold becomes part of their calendar. It prevents a warm week in midwinter from triggering premature flowering, which would be disastrous if frost returns. This is another example of experience's persistent change. The plant's history matters. What happened last month influences what happens next. Flowering is one of the most visible outcomes of timekeeping. Flowering is not just reproduction, it is timing reproduction. Flowers must appear when pollinators are active, when conditions can support seed development, and when the plant has sufficient resources. Flowering too early or too late can mean failure, so plants budget, they allocate, they choose in the biological sense among competing priorities. Resource budgeting is one of the quiet themes of plant life. A plant is always managing limited supplies. Water, minerals, light, and stored sugars are not infinite. Growth, defense, repair, and reproduction all draw from the same pool. Timekeeping helps decide when to spend, when to save, and when to take risks. A plant that senses long days and stable temperatures might invest heavily in growth and leaf expansion. A plant that senses shortening days might shift towards storage, reinforcing tissues, or preparing reproductive structures. A plant that senses drought might close stomata, reduce growth, and favour survival over expansion. All of this is guided by signals. Light signals, temperature signals, hormonal signals, internal clock signals, and it adds up to a portrait that is easy to miss if the world is viewed at human speed. A plant's experience of the world is built from slow measurements, from repeated sampling, from integration across time. It is less like watching a movie and more like reading a constantly updating set of graphs, where each line is a different environmental variable, and the plant's body is the living response. There is a quiet elegance to this. The plant is not guessing blindly. It is living inside patterns, and it has evolved to detect those patterns with a remarkable sensitivity. Even under a calm sky, the light above a leaf is changing. The ratio of red to far red shifts as neighbours grow. The intensity varies as clouds pass. The duration of day lengthens or shortens by minutes across the weeks. The internal clock ticks on, tuned by dawn and dusk. The plant adjusts, not in single dramatic events, but in continuous calibration. In that sense, plants do not simply endure time, they inhabit it, and once light and time are recognized as the primary scaffolding of plant experience, other channels begin to feel less mysterious. Air chemistry, touch, gravity, electrical pulses, hormonal waves, damage signals that spread like news, memory-like changes that alter future responses. It starts with something as simple as sunlight on a leaf. But for a plant, sunlight is never just brightness. It is direction, it is competition, it is season, it is schedule, it is warning, it is opportunity, and it is the steady, quiet clock face on which the rest of the plant's world is written. Light gives plants a sense of schedule and direction. Time gives them context. Together, those signals frame the world like a slow-moving compass and clock. But plants do not live in light alone. They live in air. Air is not empty space to a plant. It is a chemical landscape, a constantly shifting mix of carbon dioxide, oxygen, water vapor, heat, and trace molecules drifting from neighbors, microbes, and attackers. For a plant, air is both resource and message, and the boundary between those two categories is surprisingly thin. One of the most important interfaces between a plant and the air is a structure so small it can be overlooked even when it is doing something essential. The stomata. Stomata are tiny pores, mostly on the underside of leaves, each surrounded by a pair of guard cells. These guard cells are not passive valves, they are dynamic regulators. They open and close the pore by changing their internal water pressure, a property called turgor pressure. When guard cells take in water, they swell, and the pore opens. When they lose water, they relax, and the pore closes. This matters because stomata control gas exchange. Carbon dioxide must enter the leaf for photosynthesis. Oxygen and water vapor exit as byproducts of the leaf's chemistry and physics. Every open stoma is, in a sense, an invitation. Carbon dioxide, come in. Water, you may leave. A leaf can only take in carbon dioxide from the atmosphere if it keeps stomata open. But an open stoma is also a leak in the plant's water economy. Water escapes through evaporation. This process is called transpiration, and it can be enormous in total. A mature tree can move vast quantities of water from soil to air over a day, though the exact amount varies by species, temperature, humidity, wind, and soil moisture. So stomata are a balancing act. They respond to carbon dioxide concentration, they respond to humidity, they respond to light, they respond to temperature, they respond to internal chemical signals from the rest of the plant, especially signals that indicate water stress. In practical terms, a leaf is constantly negotiating. If carbon dioxide is plentiful, stomata might not need to open as widely. If humidity is low and the air is dry, the risk of water loss rises, and stomata may close to conserve water. If light is strong, photosynthesis can run faster, and carbon dioxide demand rises, which encourages opening. If a heat wave arrives, water loss can become dangerous, and closing becomes a survival strategy, even if it limits growth. This is a vivid example of experience as integration. The guard cells are not merely reacting to one stimulus. They are responding to several interacting conditions, producing an outcome that reflects a trade-off, energy gain versus water loss. And the air carries more than carbon dioxide and water vapor. It also carries chemical hints about other living things. Plants produce volatile organic compounds, often shortened in scientific writing to VOCs, though the full phrase is more voiceover friendly, volatile organic compounds. These are small molecules that evaporate easily and drift through air. Some are released continuously at low levels. Others increase dramatically during stress, damage, or infection. Many of these airborne molecules are the same kinds of chemicals humans associate with smell: the sharp scent of crushed leaves, the rich green aroma of cut grass, the resinous edge of a pine tree on a warm day. Those smells are not just human experiences, they are chemical emissions with ecological effects. When a plant is damaged, especially by insects, it can release a blend of volatiles. Some of these chemicals can attract predators or parasites of the herbivore, effectively recruiting bodyguards. In several systems, caterpillar feeding triggers plants to release compounds that draw in parasitic wasps, which then lay eggs in the caterpillars. The plant is not plotting revenge, but the chemical outcome can tilt the local ecological balance. Other volatiles can influence nearby plants. A neighbouring plant may detect certain airborne chemicals and alter its own physiology in response, for example, by priming defence pathways so it can respond faster if attacked. This is often described as plant volatile signalling, and it is measurable. It can be observed in controlled experiments with chemical analysis of the air and physiological measurements of plant responses. But it also has clear limits, and those limits are part of being honest about the science. Air is messy. Wind direction changes. Molecules dilute quickly. Environmental conditions scramble signals. Many volatile effects are short-range. Some are strong in laboratory setups, but harder to demonstrate in complex natural environments where countless chemicals are present at once. Not every smell is a message. Not every response is a conversation. Sometimes it is simply chemistry drifting and a nearby organism reacting opportunistically. Still, within those limits, the underlying point remains. Plants can detect airborne cues and adjust behaviour accordingly. This means a plant can, in a sense, read nearby vegetation. Some of that reading happens through light, as with the red to far red shifts that signal crowding. Some happens through air chemistry, volatile emissions from neighbours, changes in carbon dioxide levels near dense foliage, shifts in humidity within a canopy. A plant in a crowded environment experiences a different atmosphere in every sense. It is literally breathing a different mix of gases in a different physical microclimate, surrounded by the chemical fingerprints of other organisms. Air sensing becomes even more important under drought and heat stress. When soil dries, water becomes harder to extract. The plant responds by producing a hormone called abscisic acid, which travels and influences guard cells to close stomata. Closing stomata reduces water loss, but it also limits carbon dioxide intake, reducing photosynthesis. This trade-off can slow growth, but it protects the plant from hydraulic failure, where water transport inside the xylem breaks down. Under heat stress, stomatal decisions can shift again. In some conditions, plants may keep stomata open to increase transpiration and cool leaves through evaporation, much like sweating. But that cooling comes at a water cost. When heat and drought arrive together, the plant is forced into a harsh compromise: cool the leaf and risk dehydration, or conserve water and risk overheating and metabolic damage. Air sensing also intersects with pathogen defense. Many plant pathogens, including fungi and bacteria, enter through stomata or exploit wounded tissue. Some plants can close stomata in response to pathogen-associated molecular patterns, chemical signatures associated with microbes. This is a kind of immune-like behaviour, where the plant detects a threat and changes its boundary conditions to reduce vulnerability. So the air is not only a supply of carbon dioxide, it is a space where danger and opportunity leave traces. And plants are tuned to those traces, and if air is chemistry, then touch is physics. Touch is the moment the world stops being a distant set of signals and becomes direct contact. Pressure on tissue, strain in cell walls, bending, vibration, friction. Plants may seem fragile, but they are built to detect mechanical forces constantly. Wind, rain, passing animals, neighbouring plants, and the plant's own weight all impose physical stress. Mechanosensation is the ability to detect mechanical changes. At the cellular level, this often means sensing stretch and pressure. Plant cells have rigid walls, but they are not static. They deform. Membranes shift. Ion channels respond to tension. When a cell is stretched or compressed, proteins embedded in the membrane can change shape and open channels, altering ion flow and triggering signaling cascades. One important result is that mechanical forces can become information, not as a story, but as a measurable state change inside the plant. A classic example of touch-guided growth is thigmotropism, where plants grow in response to contact. Vines and tendrils use thigmotropism as a navigation strategy. A tendril brushes against a support, and growth patterns change so the tendril curls, tightens, and secures the plant to a structure. The plant gains stability and access to light without investing heavily in thick supportive stems. This is not a one-off reaction. The plant continues to sense tension and adjust. Tendrils can coil in ways that maintain grip and distribute stress. Some can even form spring-like structures, allowing flexibility in wind while preserving attachment. Mechanical sensing can also be rapid, which surprises many people who assume plant responses are always slow. Mimosa pudica, often called the sensitive plant, is famous for folding its leaves when touched. A light brush can trigger a cascade of electrical and hydraulic changes. Cells at the base of the leaflets lose turgor pressure quickly, and the leaf collapses inward. The response can happen in seconds. This is not a conscious flinch. It is a physical mechanism triggered by mechanosensory input and mediated by rapid signaling. The adaptive value is still debated in detail, but plausible functions include deterring herbivores, reducing leaf damage, or shaking off insects. Whatever the exact advantage in a given context, the important point is that plants can convert touch into fast, coordinated movement. Then there is the Venus flytrap, a plant that seems almost designed to break human categories. The Venus flytrap has specialized leaves that snap shut when triggered. Inside each trap are sensory hairs. When these hairs are touched in certain patterns, electrical signals are generated and the trap closes. The plant uses mechanical detection not just to respond, but to discriminate. A single touch is often not enough. The system typically requires repeated stimulation within a short time window. That reduces false alarms, such as a raindrop or drifting debris. The trap is making a simple decision rule: close only when a likely prey item is present. Again, no intention is needed. But the outcome is selective, and selection is a kind of information processing. Beyond these dramatic cases, mechanosensation shapes plant life in quieter ways that are everywhere. Wind exposure can lead plants to alter growth patterns, producing sturdier stems and changing the distribution of tissues. This phenomenon is sometimes described as thigmomorphogenesis, where mechanical stimulation influences development. A plant repeatedly bent by wind often grows shorter and thicker, investing in structural reinforcement rather than height. The plant has experienced a mechanical environment and adapted to it. Mechanical forces also inform how plants compete. In dense vegetation, leaves press against leaves, stems rub, branches collide in wind, roots encounter stones, compacted soil, and the resistance of other roots. Every contact changes growth trajectories. Touch is not just contact. It is constraint. It is guidance. It is a map of the physical world written into tension and pressure. And when mechanosensation is placed beside air sensing, a richer picture emerges. A plant is always negotiating its boundaries. At the leaf surface, guard cells open and close pores, trading water for carbon dioxide, reading humidity, heat, and chemical threat. Along stems and tendrils, mechanosensors detect contact and tension, steering growth toward support and away from damage. In rapid response species, electrical and hydraulic systems can turn touch into movement within seconds. In all plants, wind and weight shape development over weeks and months. None of this looks like animal experience, but it is experience in the biological sense: sensing, integration, response. The plant's world is not silent. It is full of signals. Some drift in the air, some press against the skin, and the plant, without eyes, without ears, and without neurons, still lives inside that information, continuously translating it into the shape of its own survival. Touch and air make the plant's world feel immediate. Pressure, humidity, chemistry, and contact are the kind of signals that arrive right at the surface. But there is another signal that never arrives as a gust or a tap. It arrives as a constant, a background force that is always present, always pulling, always shaping every drop of water and every strand of tissue. Gravity. Humans experience gravity as weight. Plants experience gravity as orientation. Not as a thought, not as a sense of dizziness, but as a persistent physical cue that must be responded to correctly, or growth becomes chaotic. A seed germinating in soil faces a basic problem. One direction contains air and light, the other contains water and minerals. A plant cannot afford to mix those up. The solution is gravitropism, growth guided by gravity perception. Gravitropism is not a single behaviour, it is a set of rules. Shoots tend to grow against gravity, a response known as negative gravitropism. Roots tend to grow with gravity, a response known as positive gravitropism. This matters even in the simplest moment of plant life. When a seed sprouts, the shoot must find its way upward, and the root must find its way downward. If the seed is rotated or disturbed, the plant corrects. It does not simply accept the new angle. Over hours and days it curves, gradually re-establishing the correct direction. The key to this correction lies inside certain specialized cells, where gravity is translated into internal movement. Many plants use statoliths. Statoliths are dense, starch-filled bodies found inside cells involved in gravity sensing. They can settle under the pull of gravity, shifting position when the plant is tilted. This settling is not a metaphor. It is physical movement, tiny bodies sliding within a cell, pressing against internal structures, triggering downstream signals. When statoliths settle, they influence how the plant redistributes auxin, the growth hormone that also plays a central role in phototropism. In shoots and roots, auxin's effects differ, which is one reason roots and shoots respond in opposite directions. In many shoots, higher auxin on one side promotes cell elongation. The side with more elongation becomes longer, and the shoot bends upward. In many roots, higher auxin can inhibit cell elongation. That means the side with more auxin grows less, while the other side grows more, and the root bends downward. This difference can feel counterintuitive until it is remembered that roots and shoots are not the same tissues solving the same problem. They are different organs with different priorities, using shared chemical tools in distinct ways. The outcome is a simple and powerful behaviour. A plant can be displaced, knocked over, buried unevenly, or twisted by wind, and it will still attempt to rebuild orientation. Gravity sensing also shapes root architecture beyond simple downward growth. A root system is not a single drill bit. It is a branching network. Roots explore soil by producing lateral roots that extend outward, curve around obstacles and adjust their angle depending on moisture, nutrient gradients, and physical resistance. Gravity provides a baseline direction, but roots overlay that baseline with other cues. Moisture gradients guide hydrotropism, chemical gradients guide chemotropism, physical barriers guide thigmotropism and mechanical avoidance. Gravity is the quiet constant that keeps exploration grounded, literally. Even the difference between a deep-rooted plant and a shallow-rooted one reflects how gravity sensing interacts with developmental programs. Some plants invest in vertical depth to access stable water reserves. Others spread laterally to capture surface nutrients and intermittent rain. Gravity sensing helps anchor these patterns, but it does not dictate them alone. It works as part of an integrated system. It is also worth appreciating that gravity sensing in plants is not a one-time decision. It is continuous calibration. A plant does not detect gravity once, decide which way is down, and then move on. Gravity is always acting, and the plant is always correcting small deviations. As the shoot elongates, its own weight bends it. As leaves grow, wind pulls. As the stem thickens, stiffness changes. The plant is a moving structure in a moving environment, and gravity is the persistent reference point. So the plant's experience of gravity is not the experience of falling, but the experience of constant alignment. A slow, steady insistence from the planet that direction matters. If gravity is a background force, electrical signaling is often the opposite. A sudden internal message that something has changed. Many people learn in school that electrical signaling belongs to animals. Neurons fire, muscles contract, brains compute. Plants are assumed to be purely chemical, slow, and silent. The reality is quieter and stranger. Plant cells maintain electrical gradients across their membranes, just as animal cells do. A cell membrane separates different concentrations of ions, and this separation creates an electrical potential. When ion channels open or close, that potential changes. These changes can act as signals. Plant electrophysiology is the study of these electrical properties and signals in plant tissues. It includes rapid electrical responses to touch, wounding, temperature shocks, and changes in light. It also includes longer-range signals that can travel through the plant and coordinate responses across distant tissues. One of the pioneers in this area was Jagadish Chandra Bose. In the late 19th and early 20th centuries, Jagadish Chandra Bose developed sensitive instruments and measured electrical responses in plants, arguing that plants show excitability and signal transmission. Some of his interpretations were debated then and remain debated now, but the central fact that plant tissues can generate and propagate electrical signals is now well supported. Electrical signaling in plants does not create a nervous system. It does not imply consciousness. It is a different kind of excitability, operating in a different anatomical context. In animal nerves, action potentials travel along specialized neurons, often insulated by myelin in many species, and synapses relay signals to other neurons or muscles. The system is built for speed and precise circuit-based control. In plants, electrical signals travel through non-neural tissues. They can propagate through the phloem, through connected cells via plasmodesmata, and through changes in ion flux that spread as waves. The routes are different, the speeds are different, the purposes are different. Still, one concept does overlap: action potentials. Action potentials in plants are rapid changes in membrane potential that can propagate along tissue. They are often slower than animal nerve impulses and are generated under different conditions, but they follow the same basic idea: a threshold-triggered electrical event that can carry information across distance. Plants also produce electrical phenomena, often described as variation potentials or slow-wave potentials, especially after wounding. These signals can spread from a damaged region to distant leaves and trigger systemic responses. This becomes especially important when a plant is injured. If an insect begins feeding on a leaf, the plant does not benefit from treating that bite as a purely local event. A local response is useful, but it can be even more useful to warn other tissues, because herbivores rarely stop after one taste. They move, they return, they spread damage. Electrical signals can participate in this internal warning system. Wounding can trigger electrical changes that propagate through the plant, followed by chemical signaling cascades that activate defense pathways in tissues that have not yet been attacked. This is systemic signaling, a whole organism response triggered by a local event. It is also one of the clearest examples of plants integrating information across their body. The plant is not reacting only where the damage occurred, it is adjusting its overall strategy. Electrical signaling also appears in rapid environmental changes. Sudden shifts in temperature, abrupt mechanical stimulation, or rapid changes in light conditions can trigger electrical responses. These electrical events can interact with calcium signaling, reactive oxygen species signaling, and hormone pathways, producing a coordinated shift in physiology. This is where the two major internal languages begin to overlap. Electrical changes can act as fast alerts. Chemical signals can carry sustained instructions. Together they allow a plant to respond across multiple timescales, immediate adjustment, followed by longer-term remodeling. The end result is coordinated defense and resource control. Defense is not free. Producing defensive chemicals, reinforcing tissues, and repairing damage consumes energy and nutrients. A plant must decide, in the biological sense, how much to invest in protection versus growth. Electrical and chemical signaling help distribute that decision across the organism, so the response matches the threat. Resource control becomes especially visible in responses to stress. If drought strikes, signals from roots and leaves coordinate stomatal closure, growth slowdown, and shifts in metabolism. If a pathogen attacks, signals coordinate stomatal behavior, local immune-like responses, and systemic readiness. From the outside, the plant looks still. Inside, it is communicating. Not with words and not with thoughts, but with changing electrical states and chemical gradients that carry meaning in the language of physiology. Gravity keeps the plant oriented in a world where up and down matter for survival. Electrical signaling helps the plant respond in a world where change can be sudden, local, and dangerous. Both are examples of the same underlying theme. Plants experience the world through measurable signals that guide their behavior, even when that behavior unfolds so slowly that human eyes mistake it for silence. Gravity gives a plant a steady reference point. Electrical signals give it speed when speed matters. Together, they reveal something important. Even without a nervous system, a plant still has ways to coordinate itself across distance. But coordination cannot rely on electrical alerts alone. Electrical shifts are often brief. They are excellent for triggering change, but not always for sustaining it. For longer-term control, plants lean heavily on chemistry. They use hormones. In everyday language, hormones are often described as mood chemicals, especially in humans, where hormones can influence emotion and behavior in ways people notice directly. In plants, that framing is misleading. Plant hormones are coordination molecules. They are chemical signals that help different tissues negotiate priorities. When to grow, where to grow, when to conserve resources, when to defend, when to reproduce, and when to wait. A plant hormone does not work like a single command. It works like a lever. It shifts probabilities and tendencies, changing how cells behave and how developmental programs unfold. Some hormones act locally, influencing only nearby tissues. Others can travel long distances through the vascular system. Often, the same hormone can produce different effects depending on concentration, tissue type, and the presence of other signals. This is why plant hormone biology can feel like a conversation rather than an instruction manual. The message is rarely a single word. It is a combination of levels, timing, and context. One of the most central hormones in plant life is auxin. Auxin is deeply involved in growth direction, branching, and tropisms. It is one of the main chemical tools plants use to translate environmental signals into physical shape. In phototropism, auxin distribution becomes asymmetric when light arrives from one side. Cells on one side elongate more than the other. The stem curves. In gravitropism, auxin redistribution also helps create curvature that reorients roots and shoots. Auxin does not simply cause growth. It helps decide where growth happens and in what pattern. It also shapes branching. Many plants show apical dominance, where the main shoot tip suppresses the growth of lateral buds beneath it. Auxin produced in the shoot tip can contribute to this suppression, interacting with other hormones and transport systems. When the tip is removed or damaged, lateral buds are often freed to grow, changing the plant's architecture. To a plant, architecture is not decoration, it is strategy. Branching determines light capture, stability, and reproductive potential. Auxin's power is amplified by the fact that it is actively transported in a directional way. Plants move auxin through tissues using transport proteins that create polar transport, meaning auxin flow has a preferred direction in many contexts. That allows auxin to act like a map maker, shaping gradients and peaks that guide development. If auxin is a hormone of direction and pattern, gibberellins are often hormones of elongation and developmental timing. Gibberellins can promote stem growth, seed germination, and flowering in many species. They help plants shift from one developmental stage to another, often in response to environmental cues like temperature and day length. A seed that remains dormant until conditions are right is not being indecisive. It is reading the world. Gibberellins are part of the chemical machinery that turns that reading into action, helping break dormancy when the time is right. Cytokinins often work in a complementary way. They are strongly involved in cell division, shoot formation, and delaying senescence, the process of aging and breakdown in tissues. Cytokinins help regulate growth balance between roots and shoots. In many contexts, cytokinins produced in roots can travel upward and influence shoot development, while auxin produced in shoots can travel downward and influence roots. This is a whole plant negotiation. The root system reports on what is available below ground. The shoot system reports on what is possible above ground. Hormones carry the messages. Then there is ethylene, a hormone famous for being a gas. Ethylene diffuses through tissues and can also move through air spaces around plants. It influences fruit ripening, leaf abscission, stress responses, and growth adjustments under mechanical constraint. Ethylene can increase when plants are crowded or when tissues are physically stressed. It can be part of why plants respond differently when packed tightly together. Ethylene is a reminder that hormones are not always slow liquids moving through pipes. Some hormones are gases, some move cell to cell, some travel through vascular routes, and many operate through complex interactions. What makes plant hormones especially important for experience is the idea of hormonal gradients. A gradient is a difference in concentration across space. Inside a plant, hormonal gradients can act like information maps. Cells know where they are in the plant, partly by the chemical environment around them. A bud experiences a certain auxin level and becomes suppressed. A root tip experiences a different balance of hormones and continues to divide and explore. A leaf experiencing stress shifts hormone levels and alters stomatal behavior. A developing flower experiences hormonal cues that coordinate its formation. A gradient is not a spoken message, it is an internal landscape. It tells tissues what kind of place they are in and what behavior fits that place. This is one reason plants can integrate information without a central control organ. When signals are encoded in gradients and transported patterns, coordination emerges across tissues. The plant becomes a chemically organized system. Hormone signaling also allows plants to connect distant events. A drying soil can influence leaf behavior through hormones produced in roots. Damage to one leaf can alter chemistry in others. Light conditions at the top of a plant can influence root growth below. This is integration across roots, stems, and leaves, mediated by traveling signals and shifting gradients. In a sense, hormones are the slow music of plant experience. They do not shout, they steer, and steering becomes especially visible when something goes wrong, because the world is not always light and water and gentle wind. It is also teeth, fungi, and injury. This is where it becomes important to speak carefully about pain. Pain is a subjective experience. It is associated with nervous systems and with specific kinds of neural processing. Plants do not have the anatomical machinery that animals use to generate pain as an experience. Using the word pain for plants can mislead because it suggests suffering in a way that biology cannot support. But rejecting the word pain does not mean rejecting the reality of damage detection. Plants detect damage with remarkable precision. A plant can detect a tear in tissue. It can detect the chemical signatures of herbivore saliva. It can detect pathogen molecules. It can detect changes in cell wall integrity. It can detect disruptions in water flow. These detections trigger signaling cascades that spread through tissues and coordinate responses. Damage detection is real, it is systematic, it is measurable. One major pathway in herbivory response involves jasmonic acid. Jasmonic acid is a hormone-like signaling molecule that becomes central when insects feed on plants. When leaf tissue is damaged, jasmonic acid pathways can activate, leading to changes in gene expression and the production of defensive compounds. These defenses can include toxins, bitter compounds, or inhibitors that interfere with insect digestion. Some plants increase the toughness of their tissues, others produce sticky latex. Others release volatile compounds that attract predators of the herbivores, turning the local food chain into an indirect defense system. The key point is that jasmonic acid helps coordinate a response appropriate to being eaten. Pathogen defense often leans heavily on a different signaling pathway involving salicylic acid. Salicylic acid is a molecule associated with plant immune-like responses, especially against biotrophic pathogens, organisms that feed on living tissue. Salicylic acid signaling can activate local defenses and systemic acquired resistance, a state where the plant becomes more prepared for future infection. The phrase systemic acquired resistance sounds grand, but the underlying idea is simple. If one part of the plant has encountered a pathogen, it may be beneficial for the rest of the plant to shift into a more defensive mode, even before the pathogen spreads. This is experience expressed as preparedness. Plants can also distinguish different types of damage. Mechanical damage, like wind tearing a leaf, may trigger some responses, but insect feeding can trigger more specific patterns. Many herbivores do not simply rip tissue randomly. They chew in characteristic ways. They introduce saliva. They expose plant cells to particular enzymes and molecules. Plants can detect these differences through receptors and signaling patterns. This allows a plant to respond differently to a storm than to a caterpillar. The defensive outputs are varied, but they tend to fall into a few broad categories. Chemical defense includes producing toxins, deterrents, and digestion inhibitors. Structural defense includes thickening cell walls, increasing lignin, producing tougher leaves, and altering growth to reduce vulnerability. Behavioral defense, in the plant sense, can include closing stomata during pathogen threat, shedding infected leaves, or altering resource allocation away from damaged tissues. And then there is communication to neighbours, which returns to the earlier theme of volatile signalling. A plant under attack may release airborne compounds that neighbouring plants can detect, leading them to prime their own defences. This is not altruism in any emotional sense. It can be a byproduct of chemical defense, and the ecological effects can still be real. In all of this, hormones are the chemistry of coordination. Auxin guides direction and architecture. Gibberellins and cytokinins regulate development and growth balance. Ethylene alters responses to stress and crowding. Jasmonic acid mobilizes defense against herbivores. Salicylic acid mobilizes defense against pathogens. These molecules act like a distributed control system. They allow a plant to respond to what is happening now and to adjust what it becomes later. And this is perhaps the most important link to the title. A plant experiences the world not through sensation as humans describe it, but through internal chemistry that carries the imprint of external reality. Light becomes gradients. Gravity becomes curvature. Dry air becomes stomatal decisions. Damage becomes defensive chemistry. Time becomes rhythmic gene expression. The world is translated into hormones and electrical shifts, into growth patterns and hardened tissues, into choices written in the language of cells. And once that translation is seen clearly, it becomes difficult to call plants passive. They are not passive, they are busy, they are informed. They are constantly coordinating themselves against a world that pushes in from every direction, even when that pushing is as quiet as sunlight or as abrupt as teeth. Plants coordinate with hormones. They defend with chemical cascades. They adjust stomata to the air and bend toward light like slow, patient compass needles. And then there is a subtler question, one that tends to unsettle human assumptions even more than a snapping Venus flytrap. Can a plant be changed by what happened yesterday? Not in the vague way that any organism is shaped by time, but in a sharper way. Can a plant's past experiences alter how it responds in the future, even when the immediate environment looks the same? In biology, that idea is often discussed using the word memory. But again, the human meaning of memory is not the one that matters here. A plant does not recall an image, rehearse a story, or replay a moment. There is no evidence for anything like that, and it would be the wrong expectation to bring into the discussion. A biological definition of memory is more practical. Memory, in this sense, is persistent internal change that alters future responses. Something happens, the organism changes internally, Later, when something similar happens again, the response is different because of that earlier change. That definition does not require a brain. It does not require awareness. It requires only that an organism can store information in its structure or chemistry in a way that affects future behavior. Plants can do that. One of the clearest concepts here is priming. Priming means that after prior exposure to stress, a plant can respond more strongly or more quickly when it encounters that stress again. The first encounter prepares the system. The second encounter triggers a faster, more robust response. This shows up in plant defense against pathogens. A plant exposed to a mild infection or certain microbial cues can enter a state where immune-like responses activate more rapidly later. This is sometimes discussed alongside systemic acquired resistance, where a local infection leads to whole plant readiness. Priming can also occur through beneficial microbes that live around roots and influence plant immunity, shifting baseline readiness without constant high-cost defense. Priming can also occur in responses to drought, heat, and other environmental stresses. A plant that has experienced drought can, under some conditions, adjust so that it reacts differently the next time water becomes scarce. The difference might be in stomatal behavior, root growth patterns, hormone sensitivity, or metabolic shifts that protect tissues from dehydration. This is not necessarily a permanent advantage, and it is not always simple. Stress history can improve resilience in some contexts and reduce growth or reproduction in others. But the important point is that the past can leave a trace that changes future behavior. How does that trace exist in a plant? One mechanism is epigenetics. Epigenetics refers to heritable or persistent changes in gene expression that do not involve changing the underlying DNA sequence. The DNA letters remain the same, but the way the DNA is packaged, marked, and used can shift. These shifts can alter which genes are turned on, when, and how strongly. Plants are especially interesting in this area because they often show strong plasticity, the ability to adjust development and physiology based on environment. Epigenetic mechanisms can contribute to that flexibility by making some responses easier to activate after a first exposure. In practical terms, epigenetics can act like a set of bookmarks. A plant does not rewrite its genetic library, but it can mark certain pages so they open more quickly next time. Drought history offers a good example of how this can matter. When soil dries, plants typically increase abscisic acid signaling, close stomata, adjust growth, and shift metabolism to conserve water. After a period of drought, some plants show altered stomatal sensitivity, meaning they may close stomata more quickly when dryness returns, or they may maintain tighter control over water loss. They may also adjust growth strategy, investing more in roots relative to shoots, or changing leaf structure to reduce transpiration. These changes can be adaptive in a world where drought is recurring, but they are not magical. They come with trade-offs. A plant that keeps stomata more tightly controlled might take in less carbon dioxide and grow more slowly under mild conditions. A plant that invests heavily in roots may allocate fewer resources to reproduction. Plant memory, in this sense, is never just advantage. It is strategy under constraint. This is where it becomes important to distinguish what is known from what is often overstated. It is known that plants show priming and persistent changes after stress exposure. It is known that gene expression and hormonal sensitivities can change in lasting ways. It is known that epigenetic mechanisms contribute to some forms of persistence, and that some epigenetic marks can last across cell divisions, and sometimes across generations in certain contexts. What is debated is how to interpret these facts. Some popular claims describe plant memory as if it were the same as human remembering, implying deliberate learning, conscious recall, or emotional experience. That is not supported by evidence, and it blurs useful distinctions. Another oversimplification is the idea that any persistent change counts as learning in a human-like sense. Biology can use the word learning in a broad operational way, but that does not mean the mechanisms are the same across organisms. A thermostat can adjust behavior based on past temperature readings, but that does not make it a thinker. In the same way, a plant can be primed and yet remain entirely without conscious experience. The careful view is that plants can store information in their physiology, they can carry forward internal states, they can respond differently because of what happened before. That is real, it is measurable, and it fits the biological definition of memory without implying the wrong kind of inner life. If a plant's memory is written in chemistry and gene regulation, then a plant's exploration is written in growth. Because plants cannot travel, they extend themselves into the environment. They send roots into soil, stems into air, leaves into light. Their movement is not locomotion, it is expansion. Nowhere is this clearer than in roots. Roots are sometimes treated as plumbing, a way to anchor the plant and suck up water. In reality, roots behave more like explorers. They are sensory organs in their own right. They detect moisture, nutrients, oxygen availability, toxins, microbes, and obstacles. They respond by changing direction, altering branching patterns, and reshaping their architecture. A root tip is a busy place. Cells divide, cells elongate, cells secrete compounds into soil, cells sense gradients. The root cap protects the growing tip and helps it push through particles, but it also participates in sensing. Roots experience the world mostly as gradients and resistances. In air, a leaf experiences changing humidity and carbon dioxide levels. In soil, a root experiences patches of nitrogen, pockets of moisture, and the mechanical resistance of compacted earth. It is a world without open space, where every millimetre is negotiated. One key concept is chemotropism in roots, growth guided by chemical gradients. Soil is not uniform. Nutrients are patchy. A decaying leaf can create a local hotspot of nitrogen. A pocket of mineral-rich material can lie beside a region that is almost barren. Roots respond to these differences by biasing growth toward richer zones. They do not need to know why nutrients are there. They only need to detect the gradient and grow into it. Moisture gradients can guide hydrotropism, where roots grow toward higher water availability. This can interact with gravitropism. Gravity tends to pull roots downward, but if moisture is higher in a particular direction, roots can curve toward it. The resulting architecture is a compromise between baseline orientation and local opportunity. Root branching is one of the most powerful tools plants have for exploration. In patchy environments, roots often branch more in nutrient-rich patches. This creates a foraging pattern. The plant invests growth where payoff is high and reduces investment where payoff is low. Again, this is not intention. It is a physiological rule expressed through hormone signaling and local sensing. This foraging behavior depends on soil physics as well as chemistry. Soil compaction matters. Compacted soil has less pore space. Roots face greater mechanical resistance. Oxygen availability drops because air cannot circulate as easily. Water infiltration changes. A root pushing into compacted soil may slow, thicken, or redirect growth. It may also alter exudates, the chemicals released into the soil that can influence microbial communities and nutrient availability. Porosity matters too. Soil with good pore structure allows roots to move more easily, allows oxygen to reach deeper zones, and allows water to infiltrate in predictable ways. A root in porous soil experiences less resistance and can explore faster, expanding the plant's reach. Oxygen availability is often overlooked, but roots need oxygen for respiration. In waterlogged soils, oxygen can become scarce because water fills pores that would otherwise hold air. Some plants respond by forming parenchyma, tissues with air spaces that help transport oxygen. Others adjust root growth to avoid suffocation. In flooded environments, roots experience the world not just as wet, but as oxygen poor, and their development reflects that. Roots also interact continuously with other organisms. They encounter fungal networks, bacteria, nematodes, and the roots of neighbouring plants. These encounters change chemistry. They change nutrient access. They change disease risk. They change growth decisions. The root is not simply absorbing, it is negotiating. And just as leaves can be shaped by light history, roots can be shaped by soil history. A plant that experienced drought may alter root depth, branching density, or sensitivity to moisture gradients. A plant that experienced nutrient scarcity may adjust how aggressively it forages. Some of this can reflect priming, some reflects long-term developmental adjustment, and some reflects the simple reality that growth itself is a record. Where a plant has grown is part of what it becomes. This is one of the quiet truths of plant life. Plants do not keep diaries, they do not replay memories, but they carry their history and their structure. A leaf is shaped by light, a stem is shaped by wind and weight, a root system is shaped by the patchiness and resistance of soil. Stomata are shaped by drought history. Defense readiness is shaped by past attack. If experience is the world leaving a trace in a living system, then plants experience the world constantly. And nowhere is that more obvious than in the underground architecture they build, centimetre by centimetre, guided by gradients, constrained by resistance, and quietly changed by everything that has happened before. Roots explore by building a map out of gradients and resistance. Stress leaves traces that alter future responses. In that sense, a plant is not only sensing the world, it is carrying the world forward inside itself. But for a plant to behave as a single organism, rather than a collection of independent parts, information has to travel. A root experiencing drought must be able to influence leaf behavior. A leaf under attack must be able to influence distant tissues. A shaded branch must be able to influence growth priorities elsewhere. Without that coordination, a plant would be a set of disconnected reactions, each part solving its own local problem without regard for the whole. The long-distance communication lines of a plant are its vascular tissues, xylem and phloem. Xylem and phloem are often introduced as transport systems, and that is correct but incomplete. They are transport systems with signaling roles. They move resources, and they move information. They are the internal roots that connect the sensory world of the leaf to the sensory world of the root. Xylem is best known for transporting water and dissolved minerals upward from roots to shoots. It is the plant's main water delivery system. Xylem flow is driven largely by transpiration, the evaporation of water from leaves that pulls water upward through a continuous column. It is not a pump in the animal sense. It is a physics-driven flow, a tension system created by evaporation and cohesion. Phloem is best known for transporting sugars produced by photosynthesis, moving energy-rich molecules from source tissues, like mature leaves, to sink tissues, like roots, developing leaves, fruits, and growing shoots. Phloem transport relies on pressure gradients created by loading and unloading sugars. Again, it is a physical system, but one managed actively by living cells. Both systems carry more than their headline cargo. Water in xylem can carry chemical signals. Phloem can carry hormones, RNA molecules, proteins, and other messengers that coordinate growth, defense, and development. One important form of signaling in xylem is hydraulic signaling. A hydraulic signal is information carried by changes in water pressure and water status. When soil dries, roots experience a drop in water availability. This changes the tension and flow in the xylem. Leaves begin to experience changes in water potential, the tendency of water to move from one place to another. Those changes can be detected quickly, sometimes faster than slower chemical signals can accumulate. In drought and heat stress, hydraulic signals can act as early warnings. A sudden rise in evaporative demand, for example during hot, dry wind, can increase transpiration and strain the water column. The plant can respond by closing stomata to reduce water loss. That response may be influenced by hormones like abscisic acid, but the immediate cue can include hydraulics, the physical status of water transport itself. This matters because it links the air and the roots in a single system. A leaf is not just reacting to the humidity above it. It is reacting to whether the plant can safely supply water to match that demand. and the leaf can only know that by receiving information from the vascular system. Chemical messengers travel through these same routes. Hormones such as abscisic acid, auxin, cytokinins, and others can move through xylem and phloem depending on context. A signal produced in roots can influence shoot behavior. A signal produced in leaves can influence root growth. This is how a plant coordinates distant tissues without a central organ. It is also how whole plant decisions emerge, especially decisions about growth versus defense. Growth is expensive. It requires water, carbon, minerals, and time. Defense is also expensive. Defensive compounds, structural reinforcement, and immune-like responses all draw from limited resources. If a plant invests heavily in defense, it may grow more slowly. If it invests heavily in growth, it may be more vulnerable. Vascular signaling helps negotiate these trade-offs across the organism. If herbivory triggers jasmonic acid signaling in leaves, that information can propagate, altering metabolic priorities elsewhere. If pathogen responses trigger salicylic acid signaling, that can shift whole plant readiness. If drought signals rise from roots through abscisic acid and hydraulics, leaves may reduce photosynthesis and growth, shifting into conservation mode. These are not deliberate choices, but they are decisions in the operational sense, outcomes selected among competing possibilities by integrating information. There are limits to vascular signaling, and it is worth stating them plainly. Many signals travel slowly compared to neural impulses. Hormone transport and chemical diffusion are not instant. Hydraulic changes can be relatively fast, but chemical adjustments can take minutes to hours to fully propagate and take effect. Yet slow signals can still be highly informative when timescales match. A plant does not need millisecond precision to respond to drought developing over days. It does not need split-second reaction time to adjust flowering based on seasonal day length. It benefits more from reliability and integration than raw speed. This is why plants often operate like systems with long memory and careful averaging. They respond to trends, not flickers. A temporary cloud does not rewrite growth strategy. A brief gust of hot air does not necessarily trigger a full drought response. Signals are interpreted in context, and context often means time. And once the vascular system is recognized as an information network, another boundary begins to dissolve, the boundary between plant and soil. Because below ground, the plant's sensory world is not limited to its own tissues. It includes an entire living zone shaped by root outputs and microbial responses. This zone is called the rhizosphere. The rhizosphere is the biologically active region of soil directly influenced by roots. It is not just dirt, it is a crowded environment where roots release chemicals, microbes respond, nutrients shift form, and interactions intensify. If the leaf surface is a boundary between plant and air, the rhizosphere is a boundary between plant and soil life. Roots constantly release compounds into the rhizosphere. These are called root exudates. Root exudates can include sugars, amino acids, organic acids, and a wide range of secondary metabolites. Some exudates are byproducts of metabolism. Others function more like deliberate outputs, shaping the environment around the root. These exudates feed microbes. They also recruit microbes. They can alter pH locally. They can bind minerals. They can influence which microbial species thrive near the roots. In other words, roots do not only passively absorb from soil, they actively engineer the soil microenvironment. Microbes, in turn, alter nutrient availability and stress tolerance. Some bacteria can fix nitrogen, converting atmospheric nitrogen into forms plants can use, often in partnership with legumes. Other microbes can solubilize phosphorus, making it more accessible. Some produce compounds that influence plant hormone pathways, indirectly altering growth. Some protect against pathogens by outcompeting them, or by triggering plant immune priming. These microbial effects can change how a plant experiences the soil. A plant root in sterile soil experiences a very different world than a plant root in a living rhizosphere. Nutrients may be present, but inaccessible without microbial help. Pathogens may be more dangerous without microbial competition. Stress tolerance may be lower without beneficial partners. This leads to feedback loops. A plant releases exudates, microbial communities shift, nutrient availability changes, plant growth and stress responses change. Those changes alter exudate patterns again. The system is dynamic. The plant is not separate from the rhizosphere. It participates in shaping it, and the rhizosphere participates in shaping the plant. These plant microbe feedback loops are a key part of plant environmental experience. They influence how plants respond to drought, nutrient scarcity, and disease. They also influence how plants compete with each other, because the rhizosphere can become an extension of a plant's strategy. In some discussions, this interconnectedness is framed using the concept of the holobiont. The holobiont is a framework that treats a host organism and its associated microbial community as an ecological unit. Applied to plants, it suggests that a plant and its microbiome together form a functional system. This concept can be useful as a reminder that organisms do not exist alone, but it can also be overstated if taken as a claim that the plant and microbes are a single fused organism in every sense. The careful view is that the rhizosphere is a partnership zone with real effects, and those effects can be described without turning metaphor into biology. What remains undeniable is that the rhizosphere is a sensory interface. A root does not only sense moisture and minerals directly, it senses the consequences of microbial activity. It senses chemical transformations happening nearby. It senses competition and cooperation through the living chemistry of the soil, and the plant's vascular system ties this underground world to the above-ground world. Signals move upward and downward. Water status, nutrient cues, hormone flows, and metabolic priorities are negotiated through xylem and phloem. The plant integrates the air, the light, the soil, and its own internal history into a coordinated whole, which means that when a plant experiences the world, it is not experiencing a simple environment. It is experiencing a world that it partly builds, a world shaped by its own chemistry, a world carried through its own internal transport lines, a world alive with microbial feedback, a world that never stops exchanging information even when nothing seems to move. Beneath the soil, the rhizosphere is already crowded. Roots release sugars and acids. Microbes respond. Nutrients change form. Threats and allies mingle in a thin, living skin around each root tip. But there is another set of partners down there that changes the entire scale of what a plant can reach. Fungi. Not the mushrooms that appear after rain, but the vast, hidden bodies of fungi that live as threads. These threads are called hyphae, and together they form a mycelium, a network that can spread through soil and weave itself into root systems. When fungi and plant roots form long-term partnerships, the relationship is called mycorrhiza. The word comes from roots meaning fungus and root, which is fitting, because this is not a casual interaction. It is an intimate interface, where fungal tissue and plant tissue exchange resources with remarkable efficiency. Mycorrhizal fungi act as symbionts, exchanging minerals for sugars. The plant is good at capturing carbon, using photosynthesis to turn carbon dioxide and light into carbohydrates. The fungus cannot photosynthesize. It needs carbon from somewhere. The fungus, however, is often excellent at exploring soil. Its hyphae can be far thinner than a root hair, slipping into tiny pores and navigating complex soil structure. Those hyphae can access minerals and water in places roots cannot easily reach. They can also release enzymes and acids that help liberate nutrients bound to soil particles. So the trade is straightforward. The fungus helps gather minerals and water, often including phosphorus and nitrogen, in forms that become more accessible. The plant pays the fungus in sugars. This exchange can reshape what experience means for a plant, because it changes the plant's reach. A root system is not only what the plant builds with its own cells. With mycorrhizal partnerships, the plant's effective sensory and resource collecting boundary extends outward through fungal tissue. That extension can be local and small, or it can become part of a much larger phenomenon. In many ecosystems, mycorrhizal networks can connect multiple plants through shared fungal partners. The hyphae associated with one plant can join with hyphae associated with another, forming networks that link roots across distances that might surprise anyone who imagines plants as isolated individuals. This is where a popular metaphor enters: the wood wide web. The phrase is memorable, and it captures something real. Forests can contain extensive fungal networks connecting roots, and those networks can transport resources and influence plant interactions. Research associated with Suzanne Simard is often linked with this idea. Suzanne Simard's work, especially in forest systems, has explored mycorrhizal connectivity and how carbon and other resources can move through fungal networks. Her research has been central to public fascination with the idea that trees may be linked underground in ways that matter ecologically. The key is to hold two ideas at once. One idea is supported by strong evidence. Mycorrhizal fungi form extensive connections, and these connections influence nutrient acquisition, water relations, and plant health. Fungal networks can move resources. They can shape competition and cooperation. They can influence which seedlings establish successfully. They can change how plants respond to stress. The second idea is where the metaphor can run ahead of the data, that forests are functioning like an intentional internet of messages, where trees deliberately send help to specific neighbours, or where information flows as if plants are consciously chatting underground. The wood wide web metaphor can be useful as a doorway into the topic, but it has limits. One limit is that networks do not automatically imply purposeful communication. A highway network allows cars to move, but it does not decide where the cars should go. In a similar way, fungal networks can create pathways for movement, but movement depends on gradients, demand, supply, and physiology, not on intention. Another limit is that fungi are not passive wires. They are living organisms with their own interests. A fungus is not simply serving the plant community, it is negotiating its own survival and growth. In many cases, mycorrhizal fungi may allocate resources in ways that benefit themselves, which may align with plant benefit, but may also create complex trade-offs. The network is not owned by the plants. It is shared with an organism that has its own agenda, even if that agenda is not conscious in any human sense. Another limit is variability. Not all mycorrhizal relationships are the same. Different fungi behave differently. Different plant species form different types of mycorrhiza. Environmental context matters. Soil nutrients, moisture, and community composition can shift whether the relationship is strongly beneficial, mildly helpful, or less effective. So what can be said with confidence? Shared networks can change what information and resources reach a plant by altering access and by buffering environmental extremes. A seedling connected to a fungal network may gain nutrients more efficiently than one relying only on its own root hairs. A plant under drought stress may receive water or nutrients more reliably through fungal pathways that explore deeper or moister microzones. In some cases, carbon movement through networks has been demonstrated, showing that resources can move between connected plants under certain conditions. Networks can also act as ecological stabilizers. They can influence which plants thrive, how quickly ecosystems recover after disturbance, and how nutrient cycles flow through a community. And even when the word information is used carefully, it can still be meaningful. A plant linked to a fungal network may receive chemical cues, microbial signals, or changes in nutrient flow that reflect what is happening to its neighbours. If a neighbouring plant is heavily drawing phosphorus, that can alter local availability. If a pathogen changes root chemistry in one plant, microbial communities and chemical conditions can shift around connected roots. These changes are not messages in the human sense, but they are information in the biological sense, patterns that can influence response. Seen through the lens of plant experience, mycorrhizal partnerships expand the plant's world. They extend sensing into soil microspaces the plant could not access alone. They reshape the plant's nutrient landscape. They modify stress response. They shift the meaning of the boundary between self and environment. A plant that is deeply partnered with fungi is not experiencing soil as a simple pool of minerals. It is experiencing soil through a living interface that can amplify opportunities and soften hazards. That brings the topic back to the surface, to a different kind of interface between organisms. Because plants do not only connect underground, they also influence each other above ground, through the air, and through shared cues. And this is where the line between what is real and what is myth needs to be drawn with particular care. The idea that plants signal to each other is not pure fantasy. There is real science here, but it is an area where popular descriptions often become too confident, too tidy, and too human. A grounded starting point is airborne signaling using volatile compounds released during herbivore attack. When an insect feeds on a leaf, the plant can release a blend of volatile organic compounds into the air. These compounds can be detected by other organisms. Some attract predators of the herbivore. Some can alter microbial communities on leaf surfaces. Some can be detected by neighbouring plants, which may respond by changing gene expression and increasing readiness for defence. This can look like communication, and in an operational sense it can be described that way. One organism releases a cue, another organism responds. But careful language matters, because the mechanism is chemical ecology, not conversation. Often, the most accurate framing is that plants emit cues, and other plants can use those cues. This leads naturally to the idea of eavesdropping, plants responding to cues produced by neighbouring plants. Eavesdropping is a useful term, because it avoids implying cooperation. It suggests that a plant can benefit by paying attention to what is already in the environment, without needing to assume that the neighbour intended to send a helpful warning. If a nearby plant is attacked and releases volatiles, a plant that detects those volatiles and primes its defences might gain an advantage. That advantage does not require the attacked plant to be altruistic. It only requires that volatile release occurs and that detection is possible. But how strong is this effect in real ecosystems? This is where experimental challenges matter. In laboratory conditions, it is easier to show cause and effect. Plants can be placed in controlled airflows. Volatiles can be measured precisely. Herbivores can be introduced in predictable ways. Responses can be tracked with gene expression markers and chemical assays. In the field, conditions are more complicated. Wind is variable. Chemical backgrounds are complex. Temperature and humidity fluctuate. Many plants and microbes emit overlapping compounds. Herbivory is patchy and episodic. It becomes harder to attribute a response to one specific airborne cue. This does not mean airborne signaling is not real. It means that ecological realism is difficult, and results need replication across contexts. It also means that effect sizes can vary widely. Some interactions may be strong at short range and weak at longer range. Some may depend on specific plant species pairs. Some may occur only under certain environmental conditions. A related topic that often appears in public discussions is kin recognition. Kin recognition evidence exists in some plant systems, where plants appear to behave differently when grown near genetically related individuals compared to unrelated ones. This can show up in root allocation patterns, competition intensity, or resource-sharing tendencies. But the word recognition can mislead if it is heard as a conscious act. Mechanistically, what is likely happening is detection of cues correlated with genetic similarity. Those cues might involve root exudate profiles, microbial community signatures, or other chemical markers in the immediate environment. A plant does not need to know who its sibling is. It only needs to respond differently to certain chemical contexts that tend to occur when relatives are nearby. This is a recurring theme in plant experience. Complex outcomes can emerge from simple detection rules. A plant can alter root growth when the local chemical environment shifts. That shift might correlate with the presence of kin. The outcome looks like social behaviour, but the mechanism is chemical and physiological. And once again, this is where confounds and replication become central. In experiments on plant signalling and kin effects, it can be hard to separate direct plant-to-plant cues from microbial mediation. Soil microbes can change rapidly, and they can carry signals and influences of their own. A plant's exudates can shape microbes. Microbes can shape another plant, and the chain of influence can look like direct communication when it is partly indirect. Another challenge is interpretation bias. Humans are naturally tempted to translate plant behavior into familiar social narratives. Warning, sharing, helping, cooperating, punishing, remembering. Those narratives are attractive because they make plants feel relatable. The danger is that they can flatten complexity into a story that feels true, even when evidence is partial. So, what are the strong findings that can be stated plainly? Plants emit volatile compounds that influence other organisms, including other plants under some conditions. Plants can prime defensive responses after exposure to certain cues, including cues associated with herbivory. Plants can respond to the presence of neighbours through changes in light quality, air chemistry, and a root zone chemistry. Some plants show differential behaviour in the presence of genetically related versus unrelated individuals, though the mechanisms and generality vary across species and contexts. Now, what are the popular oversimplifications that should be left behind? That plants are chatting in a human-like way, exchanging deliberate warnings as if they are a community of conscious speakers. That the Wood Wide Web is a benevolent information internet run by trees for mutual aid at all times, rather than a complex ecological network with variable outcomes and multiple competing organisms. That plant signalling always implies cooperation, rather than often being a byproduct of stress chemistry that others can exploit. Leaving those oversimplifications behind does not make the topic less interesting. It makes it more honest, and, in a quiet way, more strange. Because the real story is that plants live inside a world thick with cues, and those cues travel through air, through soil, through fungi, and through the plant's own vascular roots. Plants do not need intentions to influence each other. They only need chemistry. They only need shared space. They only need overlapping lives. A forest is not silent. It is not merely still. It is full of exchange. Sugars trading for minerals through mycorrhizal fungi. Water tension shifting through xylem. Hormones drifting through phloem. Volatiles lifting from leaves into air. Roots leaking exudates that reshape microbial worlds. Neighbours changing the colour of light simply by existing. In that landscape, plant experience becomes something larger than an individual plant standing alone. It becomes a lived ecology. A plant experiences not only sunlight and gravity, not only drought and damage, but also the presence of others, woven into its environment as chemistry, as competition, as partnership, and as the faint but persistent traces of life moving through shared networks. A plant does not get to pause and think. It does not get to step away from its environment and come back with a better plan. It grows in place, and every day it is forced to manage limits. That is a useful way to approach the idea of decision-making in plants. Decision-making, in an operational sense, can be defined as selecting outcomes under constraints. When several responses are possible, each with costs and benefits, the organism produces one outcome rather than another. There is no need to imagine intention or awareness. The definition can stay entirely grounded in physiology. Plants are constantly selecting outcomes because plants are constantly allocating resources. A plant's budget is not unlimited. Carbon captured by photosynthesis, water moved through xylem, minerals gathered by roots, and energy stored in tissues all exist in finite quantities. Those resources can be spent on growth, stored for later, invested in reproduction, or diverted into defense and repair. The same unit of carbon cannot become a new leaf and a defensive toxin at the same time. The same water cannot be used for rapid growth and also conserved indefinitely. So the plant's life becomes a continuous trade-off. Growth is the most visible option. It means building more leaf area for photosynthesis, extending roots to reach water and nutrients, thickening stems for support, and expanding overall size. Growth can be the difference between thriving and being shaded out. It can also be the difference between reaching a water pocket and drying out. But growth carries risk. Fast growth usually increases water demand. It can create taller, thinner stems. It can produce leaves that are efficient in good conditions, but vulnerable in bad ones. Growth is a bet that the environment will stay supportive long enough for the investment to pay off. Storage is a quieter option. It means holding carbon and nutrients in places like roots, stems, bulbs, tubers, or woody tissues. Storage can protect a plant through winter, drought, fire, and repeated herbivory. It can support regrowth after damage. It can buffer against unpredictable seasons. But storage also has a cost. Stored resources are not being used to capture light right now. They are not producing flowers right now. They are not building a canopy that outcompetes neighbors right now. Storage is a bet in the other direction, that survival through hardship is more valuable than immediate expansion. Reproduction is another investment with its own timing problem. Making flowers, fruits, and seeds can be extremely expensive. It often requires a plant to redirect carbon, nitrogen, and water away from vegetative growth. Yet reproduction is how a plant's lineage continues. The decision is rarely whether to reproduce at all, but when, how much, and under what conditions. Many plants solve this with time-keeping cues. Photoperiodism and temperature history influence when flowering begins, when seeds mature, and when dormancy should occur. Reproduction becomes coordinated with the environment, so that effort is spent when success is more likely. Defense is the fourth major sink for resources, and it is easy to underestimate. Defensive chemistry can be complex. Structural reinforcement can be heavy. Immune-like responses can be metabolically demanding. Repair after damage requires building new tissue and sealing wounds. All of this costs carbon, minerals, and energy. Yet defense can be the difference between being eaten alive and surviving intact. The difficult part is that defense also reduces growth, and growth can be essential for competing. This is why plants often shift priorities depending on what signals are strongest. In a safe environment, a plant can emphasize growth. In a threat-rich environment, a plant may emphasize defense. In a drought, a plant may emphasize conservation and repair. In a short growing season, a plant may emphasize rapid reproduction. So, how does a plant prioritize? It integrates signals. Signals arrive from light through photoreceptors. Signals arrive from the air through stomatal sensing of humidity and carbon dioxide. Signals arrive from roots through water status, nutrient gradients, and microbial interactions. Signals arrive from damage detection pathways triggered by herbivores or pathogens. Signals arrive from internal clocks that track time of day and season. Signals arrive from the plant's own hydraulics, reflecting whether water transport is safe or close to failure. These signals converge on hormone networks, gene regulation, electrical and calcium signaling, and resource availability. The outcome is not a single command. It is a shifting balance where certain pathways become more active and others are suppressed. Under stress, this balancing becomes obvious. In drought, abscisic acid signaling increases, stomata close, growth slows, and metabolism shifts toward survival. In herbivory, jasmonic acid signaling can rise, defensive chemistry increases, and growth may be temporarily deprioritized. In pathogen pressure, salicylic acid signaling can activate immune-like responses that change resource allocation and sometimes restrict growth. The plant is not deciding in a human sense. It is selecting outcomes because its internal networks weigh competing demands. This is why complex behavior can emerge without intention or awareness. A plant can respond differently to the same light level depending on water availability. It can allocate more resources to roots when nutrients are scarce. It can prime defenses after a first attack. It can delay flowering until seasonal cues align. These are sophisticated outcomes. They are also entirely explainable through physiology. This is one reason the topic has attracted debate around plant behavior and intelligence. Stefano Mancuso is often named in these discussions, arguing that plants show rich behavioural repertoires and that intelligence can be framed more broadly than brain-based cognition. Whether one prefers the word intelligence or not, the useful takeaway is that plants deserve to be treated as active systems that sense, integrate, and respond in ways that can be complex, adaptive, and context-dependent. The most accurate framing is not that plants are thinking. The most accurate framing is that plants are continuously solving problems with the tools they have: hormones, electrical signaling, gene regulation, hydraulic control, and a body plan built for slow, distributed adaptation. Once that is clear, the next pressure becomes unavoidable. Plants do not experience their environment alone. They experience it in the presence of neighbours. Competition is not just an event. It is a condition. In a meadow, a forest, a crop field, or a crowded garden bed, nearby plants reshape the environment simply by existing. They absorb light, pull water, extract minerals, alter airflow, and modify the microbial world around roots. What matters is that plants can detect this early, often before physical contact occurs. One of the most powerful early signals is the red to far red ratio of light. Sunlight contains a mix of wavelengths. Leaves absorb red light efficiently for photosynthesis and tend to transmit or reflect more far red light. When vegetation becomes denser, the light environment shifts. Red light becomes relatively scarcer. Far red becomes relatively more prominent. This shift can occur even before a plant is fully shaded, which makes it an unusually useful cue. Phytochromes detect this change, and in many species it triggers shade avoidance syndrome. Shade avoidance syndrome is not a single response. It is a coordinated set of growth changes that often include stem elongation, altered leaf angles, and changes in branching. The plant reallocates resources toward reaching brighter light, essentially treating the spectral shift as a forecast of future competition. This is how plants detect crowding before leaves touch. They do not need a collision to know the canopy is filling in. They can read the light. Competition, however, is never only about light. A dense stand of plants also intensifies competition for water and nutrients. After rain, soil moisture is drawn down faster when many roots occupy the same volume of soil. Nutrient patches are depleted more quickly. Roots respond by altering their architecture, branching more aggressively in rich zones, extending deeper when surface layers dry, or shifting growth away from compacted or oxygen-poor pockets. These adjustments are guided by gradients of moisture and chemistry, but they are sharpened by the presence of other root systems extracting the same resources. Neighbour sensing also interacts with risk. The shade avoidance strategy can help a plant reach light, but it can reduce stability and resilience. A plant that elongates rapidly may become taller and thinner. Thinner stems can be more prone to bending under wind or rain. Rapid growth can increase water demand, which becomes dangerous if drought arrives. Investment in height can also reduce investment in roots, making the plant less secure in both soil and water supply. So competition forces a gamble. Stretching for light can win the race upward, but it can also make the plant more fragile. Avoiding elongation can preserve stability and water balance, but it can lead to being shaded and starved of energy. Different species solve this in different ways. Some commit to shade avoidance and speed. Others are shade tolerant, investing in leaf physiology that captures light efficiently under low intensity. Some mix strategies depending on life stage and environmental context. Even the air changes with density. Crowded foliage alters airflow and humidity, which can affect transpiration rates and pathogen spread. In humid, still air, fungal diseases can become more likely. That shifts defensive priorities. The plant's experience of neighbours includes not only resource competition, but also changes in microclimate and threat landscape. The most grounded way to think about neighbours from a plant's perspective is as predictable shifts in signals. Light quality changes as leaves filter sunlight. Water availability becomes more contested as roots overlap. Nutrient gradients become more transient as multiple plants draw from the same patches. Root zone chemistry changes as exudates and microbes respond to multiple hosts. Airflow and humidity shift inside dense vegetation, changing water loss and sometimes disease risk. These are not stories, they are measurable conditions. And because they are reliable, plants have evolved reliable responses. A plant does not need awareness of competitors to behave competitively. It needs sensors that detect the environmental fingerprints of crowding and internal systems that reallocate growth, storage, and defense accordingly. That is what competition looks like when it is experienced through plant biology. Not as rivalry in the human sense, but as a constant shifting of light, water, nutrients, and risk, written into the signals a plant can detect and expressed through the growth patterns it cannot avoid producing. A plant can handle competition, but competition is only one part of the environment's pressure. Even a solitary plant, miles from the nearest neighbour, still lives inside forces that do not negotiate. Heat, cold, drought, salt. These are not rare events in plant life. They are common conditions that arrive in waves, sometimes predictable, sometimes sudden. And because plants cannot walk away, stress becomes a kind of long-term relationship. The plant measures it, adjusts to it, and carries it forward in its physiology. Drought is one of the clearest examples of stress as lived experience, because it forces a plant to manage a simple but unforgiving equation: water loss versus carbon gain. Water leaves a plant mainly through stomata. Carbon dioxide enters mainly through stomata. Open stomata feed photosynthesis and growth, but open stomata also leak water into dry air. Close stomata conserve water, but close stomata slow photosynthesis. This trade-off is managed through a hormone that appears again and again in plant stress biology: abscisic acid. Abscisic acid is a major drought response hormone and a key stomatal regulator. When soil dries, roots experience water shortage and internal water status shifts. Abscisic acid levels rise, and guard cells respond by reducing turgor pressure, which closes stomata. This conserves water by reducing transpiration. Closing stomata is not merely shutting a door, it changes the entire internal economy of the plant. Carbon dioxide intake drops, so photosynthesis slows. Sugars are produced more slowly. Growth slows. The plant shifts from expansion to survival. Abscisic acid also influences other drought adaptations. It can alter gene expression, promote production of protective molecules that help cells tolerate dehydration, and shift growth patterns so more investment goes into roots rather than leaves. In many plants, drought leads to reduced leaf expansion, thicker cuticles, and changes in root architecture that seek deeper water sources. Drought is not experienced as a single moment. It is experienced as continuous adjustment. Stomata opens slightly in the morning when humidity is higher, then close as the air dries. Roots extend toward damp microzones. Metabolism shifts toward conserving water and protecting tissues. Heat stress arrives differently. Heat can damage proteins, disrupt membranes, increase evaporation, and push photosynthesis toward inefficiency. It can also drive rapid water loss, making drought more severe even when soil moisture is not yet critically low. One of the most important protective tools plants use under high temperatures is heat shock proteins. Heat shock proteins are molecules that help protect other proteins. When proteins are exposed to heat, they can unfold or misfold, losing function. Heat shock proteins act like cellular chaperones, stabilizing proteins, helping them refold correctly and preventing damaging clumps from forming. This is not a luxury, it is a survival mechanism because a cell full of misfolded proteins can fail quickly. Heat also affects membranes, the thin barriers that keep cellular chemistry organized. Membranes must remain flexible enough to function, but not so fluid that they leak. Under heat, membranes can become too fluid, compromising transport systems and signaling. Plants adjust membrane composition to maintain function, altering the types of lipids used so the membrane remains stable under high temperatures. Heat stress also interacts with the physics of water transport. Under intense heat and low humidity, transpiration demand rises sharply. The plant may open stomata to cool leaves through evaporation, but this can threaten water status. If the water column in xylem becomes too strained, air bubbles can form, a process called cavitation, which blocks water transport. Avoiding cavitation is one reason plants can become extremely conservative with stomatal opening during combined heat and drought. Cold stress presents a different set of challenges. Cold slows chemical reactions, making metabolism sluggish. But the more dangerous problem, especially near freezing, is water behavior. When temperatures drop, water can freeze outside cells. Ice formation outside cells draws liquid water out of the cell, dehydrating it. If ice forms inside cells, it can be physically destructive, puncturing membranes and disrupting structure. This is why freezing stress often becomes a story about membranes and water behavior. Plants that tolerate freezing often adjust their internal chemistry to reduce damage. They can accumulate solutes, such as sugars and certain amino acids, which lower the freezing point of cellular fluids and help stabilize proteins and membranes. They can also alter membrane lipid composition, so membranes remain functional and less brittle at low temperatures. Many temperate plants also undergo cold acclimation. Exposure to gradually cooling temperatures can trigger gene expression changes that increase freezing tolerance. This is another example of experience's persistent change. The plant's past conditions influence its future resilience. Dormancy is also related to cold stress. Many trees and perennials enter dormancy to avoid vulnerable growth during winter. Bud scales protect developing tissues. Metabolism shifts. Growth pauses. In spring, dormancy breaks only after a combination of cold exposure and warming conditions aligns with seasonal cues. Then there is salinity stress, which can be deceptively harsh. Salt stress is not only about toxicity, though toxicity matters. High salt levels can damage cells, disrupt enzyme function, and interfere with nutrient balance. Sodium ions, in particular, can accumulate and displace essential ions like potassium, which plants need for many cellular processes. But salinity is also a water access problem. Water moves from regions of higher water potential to lower water potential. Salty soil has low water potential because dissolved ions bind water and make it harder for plants to extract. Even if soil looks wet, a plant may struggle to pull water in if salinity is high. This creates a situation that feels to the plant's physiology like drought, even in the presence of water. Plants respond by regulating ion transport, sequestering salts into vacuoles, excluding ions at roots, and producing compatible solutes that help maintain cellular water balance. These strategies cost energy. They require active transport and metabolic shifts. Salt tolerance is not a simple trait. It is an ongoing management problem. Heat, cold, drought, and salt all share a theme. They force constant measurement and adjustment. A plant must monitor its water status, membrane stability, protein integrity, ion balance, and energy budget. It must respond at multiple timescales: immediate stomatal changes, slower hormonal shifts, longer-term growth adjustments, and seasonal developmental decisions. Stress biology is the science of these responses, but from the plant's perspective, it is simply life as a continuous negotiation with physics and chemistry. When these stresses are stacked together, the plant's experience becomes even more complex. Heat often increases drought stress. Cold can amplify damage when tissues are already weakened. Salt creates drought-like conditions while also introducing toxicity. A plant's internal systems must integrate these signals and produce a coherent response. None of this looks like thought, it looks like regulation. But regulation is how plants live. And after moving through all these mechanisms, it becomes possible to step back and ask the larger question again. What does the world look like to a plant? Not visually and not emotionally, but biologically. A plant's world is a world of gradients. Moisture is not a yes or no condition. It is a gradient through soil, changing millimeter by millimeter. Nutrients are not evenly spread. They occur in patches and hotspots. Salinity can rise near the surface as water evaporates, leaving salts behind. Oxygen availability varies with soil porosity and waterlogging. Light is a gradient too. Intensity shifts with canopy cover, time of day, and cloud patterns. Colour balance shifts with vegetation density, changing red to far red ratios that signal crowding. Day length shifts across seasons, changing by minutes and then hours as the year turns. Temperature is a gradient. Leaves can be warmer than air under strong sun. Soil can be cooler than air at night. Heat waves and cold snaps alter the baseline, but microclimates exist within canopies and near the ground. Pressure and resistance shape everything. Wind bends stems. Soil compaction blocks roots. The plant's own weight creates mechanical strain. Touch becomes guidance. Vascular tension becomes warning. And chemistry is everywhere. Carbon dioxide levels shift locally around dense foliage. Humidity shifts inside canopies. Volatile compounds drift from damaged leaves. Root exudates shape microbial communities. Hormones create internal gradients that act like maps for development. Electrical changes act as alerts that something has happened. From this perspective, plant experience is less like a rapid stream of snapshots and more like slow integration over time. Animal experience is often dominated by fast sensing. Eyes, ears, quick movement, immediate threat response. Animals sample the environment in bursts and can relocate to change conditions. Speed is often survival. Plant experience is dominated by continual measurement, not a burst, but a steady reading. A plant cannot relocate, so it must adjust its internal state and its growth patterns to match the environment it cannot escape. Time is not just a background. Time is part of the sensing itself. Slow does not mean simple. Slow can mean deeply integrated. A plant may take hours to bend toward light, but that bend reflects photoreceptors, auxin transport, cell elongation mechanics, and ongoing correction as the light changes. A plant may take days to adjust root architecture, but that architecture reflects gradients of water and nutrients, microbial interactions, and the physics of soil structure. A plant may take weeks to prepare for winter, but that preparation reflects photoperiod, temperature history, gene regulation, and stored resource budgeting. When this sensory world is connected to ecology, it becomes easier to see why plants shape landscapes so strongly. In forests, competition for light and the influence of mycorrhizal fungi create layered canopies and complex underground networks. In fields and grasslands, rapid seasonal growth, grazing pressure, and patchy soil nutrients produce shifting mosaics of plant strategies. In deserts, drought and heat produce extreme water conservation physiology and timing strategies that depend on rare rainfall. In wetlands, oxygen availability and waterlogging force plants to manage root respiration and microbial relationships differently. The plant's sensory world is not abstract. It is the real set of forces that shape where plants can live and what they can become. And that returns calmly to the title. How plants experience the world is not a claim about plant consciousness. It is a claim about what plants can detect, how they integrate information, and how their bodies change in response. Plants experience the world as patterns that can be measured: light quality and day length, moisture gradients and water tension, heat and cold shaping proteins and membranes, salt altering water access and ion balance, chemical cues from neighbours, microbes, herbivores and pathogens, slow adjustments in growth, structure, and internal chemistry that carry the imprint of external reality. A plant does not witness the world the way an animal does, but it is continuously informed by the world, and it continuously becomes, cell by cell, the living record of what it has measured. Plants experience the world through signals: light, humidity, gravity, chemistry, pressure, time. Those signals become internal shifts in hormones, electrical states, gene expression, and growth. The plant responds, and the response becomes the plant's body. On its own, that might sound like a private story, a leaf responding to shade, a root turning toward moisture, a stem thickening after wind, a tree closing stomata during drought. But plants do not exist in isolation. When plants sense and respond, the consequences do not stop at the edge of a leaf. They scale outward, from individual physiology to ecosystems, from ecosystems to the atmosphere, from the atmosphere to climate patterns, and from climate patterns back to the conditions plants must sense again. This is the quiet consequence, the way plant experience shapes Earth. The most obvious example is oxygen. Earth's oxygen-rich atmosphere is not guaranteed by physics. It is a biological outcome. Oxygen is produced primarily through oxygenic photosynthesis, the process in which plants, algae, and cyanobacteria use light energy to split water and move electrons through photosynthetic machinery, releasing oxygen as a byproduct. On land today, plants are a major contributor to the global oxygen cycle, not because they are trying to make air breathable, but because they capture carbon and, in doing so, release oxygen. The planet's atmosphere is partly the exhaust of plant metabolism. This links directly to how plants experience light. A leaf's photoreceptors tune growth and development, but the deeper engine is photosynthesis itself. Photosynthesis depends on light intensity, temperature, water availability, and carbon dioxide concentration. A plant's ability to sense and respond to those conditions affects how much carbon it fixes, how fast it grows, and how much oxygen is released in the process. Oxygen is one result, carbon cycling is another. Plants are one of Earth's major pathways for moving carbon from the atmosphere into living tissue. Carbon dioxide enters leaves through stomata, becomes sugars, becomes cellulose, lignin, starch, roots, trunks, seeds, and entire forests. Some of that carbon returns quickly to the air through respiration and decay. Some enters soils as litter and root material. Some becomes long-lived woody biomass. Some becomes part of soil organic matter that can persist for years, decades, or longer. The details vary hugely across ecosystems, but the principle is stable. Plant sensing and response influences how carbon moves through the planet. When a plant experiences drought, it often closes stomata. That reduces carbon dioxide intake. Photosynthesis slows. Carbon uptake from the atmosphere decreases. When drought is widespread across a region, this becomes a measurable shift in ecosystem carbon exchange. When temperatures rise, plants may increase respiration, and photosynthesis may become less efficient under heat stress. When cold arrives, growth slows, and carbon allocation shifts towards storage and survival. When salinity rises, plants spend energy managing ions and may grow more slowly, altering carbon capture and biomass accumulation. All of these stresses are local experiences for a plant. Together, they become planetary feedbacks. Climate feedback is not always intuitive, but the logic is straightforward. Plants influence climate because they influence greenhouse gas exchange, water cycling, and surface energy balance. And plants change their behavior based on the conditions they detect. One of the most important bridges between plant experience and climate is stomatal behavior. Stomata regulate carbon dioxide intake and water loss. When stomata are open, plants transpire water. Transpiration moves water from soil into the atmosphere as vapor. This is not just a plant-level process. It is a significant component of the water cycle, especially in forested regions. Transpiration helps cool leaves through evaporation, but it also cools the surrounding air and contributes to humidity. In many landscapes, plant transpiration can influence local climate patterns by affecting temperature, humidity, and the likelihood of cloud formation. A forest can act like a moisture pump, moving water upward and shaping rainfall patterns downwind. When plants experience drought and close stomata, transpiration drops. The air becomes drier, cooling decreases, local temperatures can rise, the boundary layer around vegetation changes. Under severe drought, reduced transpiration can contribute to heat extremes at the surface, which can further stress plants. This can become a reinforcing loop. The precise outcomes vary with region and vegetation type, but the principle remains: stomatal decisions scale up. This is one reason plant experience is not merely an academic curiosity. It is part of how Earth regulates itself. Plant decisions about growth versus defense also scale into ecosystem stability. In a stable environment with adequate water and nutrients, plants can invest in growth, building dense canopies, deep roots, and large biomass. That structure supports entire communities: insects, birds, mammals, fungi, and microbes. It stabilizes soil, reduces erosion, and shapes microclimates. Under persistent stress, plants may shift toward survival modes: slower growth, increased defense chemistry, changes in leaf traits, altered root allocation, and sometimes reduced reproduction. Across an ecosystem, these shifts change the food web. Leaves may become tougher and less nutritious. Growth rates slow. Carbon storage changes. Fire risk can change if vegetation dries, or if species composition shifts toward more flammable forms. Even small differences in plant sensory tuning can matter. If one species closes stomata quickly under drought and another keeps them open longer, those strategies influence which species dominate over time. Dominance shifts change canopy structure, shading, soil moisture retention, and microbial communities, and the ecosystem can move toward a new equilibrium. This is ecological stability emerging from individual physiology multiplied across millions of plants. Understanding plant experience matters for agriculture, for the same reason it matters for ecosystems. Plants respond to signals, and those responses determine outcomes humans care about. Crop yield depends on timing of flowering, grain filling, water use efficiency, nutrient uptake, and resistance to pests and pathogens. Every one of those traits is tied to sensing and response. If stomata close too aggressively under mild drought, photosynthesis drops and yields can fall. If stomata remain too open under severe drought, crops can suffer hydraulic failure and die. If a crop cannot sense pathogen cues effectively, disease spreads. If it invests too heavily in defense chemistry in low-threat environments, growth and yield can decline. Agriculture is, in part, the art of managing the signals plants receive. Irrigation, fertilization, planting density, pest management, and timing. Modern plant science, breeding, and biotechnology often focus on tuning these response systems so crops can maintain productivity under stress. Climate resilience intensifies this need. Heat waves are becoming more relevant in many regions. Drought variability is a growing challenge. Salinity can increase in irrigated lands, where salts accumulate over time. Late frosts can damage crops that break dormancy too early. All of these pressures intersect with plant sensing and timing. A plant that experiences the world through signals can only thrive if its signals remain reliable. Climate change, in many ways, is a disruption of reliability. It changes the timing and intensity of cues. It can create mismatches between photoperiod, temperature, rainfall, and biological interactions such as pollinator activity or pathogen seasons. Building resilience means understanding which signals a plant trusts, how it integrates them, and how those integrations can be adjusted without breaking the plant's overall function. And after all of this, the quietest and most grounded conclusion is also the largest. Humans live inside environments continuously interpreted by plants. The air you breathe is shaped by photosynthesis and the global carbon cycle. The water cycle is influenced by transpiration and stomatal behavior. Local climates are softened or sharpened by the presence of vegetation. Soils are stabilized by roots and enriched by organic matter. Entire ecosystems are built on plant growth strategies that respond to signals: light, moisture, temperature, pathogens, neighbors. Plants do not experience the world the way humans do, but their experience, in the biological sense, is woven into the planet's functioning. Every day leaves measure light and air. Roots measure moisture and chemistry. Stomata open and close. Hormones redistribute, electrical signals ripple, growth angles shift by fractions, defensive pathways rise and fall, and over time those countless small adjustments become forests, fields, wetlands, grasslands, and the living structure of Earth. It is easy to miss because it happens quietly. But the world is not only shaped by what humans notice, it is shaped by what plants detect.